Hello, everyone. Welcome back to Creation Myths. I'm really excited for tonight. So I've been looking forward to this one since the topic that we're talking about appeared on the internet, which at this point was a while back. And I finally got around to doing my due diligence and watching this video and taking notes on it and stuff just recently. So here we are. So I just need to make sure that everything looks okay on my end in terms of YouTube. It does, other than the fact that I am blurry. Not as blurry as I was that one time, but it's not good. I promise I'll fix that eventually, but the important thing is, what we're saying, plus we're going to put a video on screen, so it's okay. As always, folks, if you notice any tech issues as we're chatting, just let me know in the chat, and I will do my best to address it. More likely, I will just flail, but, I can try. So joining me tonight to discuss Dr. Rob Carter and the crimes, his criminal enterprise, his crime spree against population genetics is my friend, my co-author, Dr. Zach Hancock. Zach, how are you doing, my friend?
I'm doing pretty well. It's been a long time now since I've been on YouTube. It's been a long time since I put out any videos. I think the last time I was live was actually on Rebecca's channel. So it's been a little bit of time. So people will notice my background is different. I'm just sitting on my couch. Sorry for the I don't know, lack of professionalism in my current setting. But I'm really excited to be here. Really excited to be talking about this topic.
Me too. And we got a super chat here from Tad is Mad, and I'm from New Jersey, so I pronounce those two things differently, which is weird. Most people would pronounce them the same, but New Jersey accent, they're actually different, which is a weird thing. But for $2 it says, love to discuss Rob Carter's many crimes. Number one, lies. Yeah, we'll be getting there. Oh, sorry, I didn't mean to click that. The chat moved on me. All right. So what we're doing tonight, folks, is Dr. Rob Carter, Creation Ministries International. I would say recently, but at this point, I think it was back in September, I want to say.
That's when the article was put out.
Oh, the article, right. So he did an article on creation.com, and then he released a video as kind of a companion to that article, kind of going through the same beats. And we're going to talk about it. So this was all about mitochondrial Eve and the dating or lack thereof for the mitochondrial most recent ancestor. The video that we are going to be viewing, not in its entirety because it was like 43 minutes long. It was called When Did Eve Live? Can We Know from Science? And the answer to those questions is approximately 1 to 200,000 years ago, and yes. Which are not the answers that Dr. Carter is going to provide us. Before we get into it, I just want to see, Zach, do you have any 30,000-foot thoughts that you want to just kind of frame this with before we get into the nuts and bolts?
Yeah, I mean, I'm sort of the unfortunate thing, I think, is that Carter, as we were talking about this a little bit before we went live, is that Carter gets so close to so many things. He's very close to seeming like he understands this stuff, and then he just takes this it's like the meme where the car immediately goes off to the exit. It's like Carter, yeah. you were almost there, and then you just missed it. So a lot of this are going to be close misses. And he has a little bit more nuance within the article than what he's going to give in the video. That's almost always the case, and I guess to some extent that's understandable. But I'm not going to let him get away with some of these little mistakes because I think they're very important, and they give misleading. They can mislead the audience if you don't pick up on them, and so these slight little deviations that he does, I'm gonna pick up on. But from the bird's eye view, from really far away, it's very sad to see Carter get so close and then just miss the target.
So Zach just gave everybody homework. Somebody make the exit ramp meme where it's understanding mainstream population genetics and mitochondrial Eve 6,000 years ago, and Carter just.
Yeah.
I want to I want to recognize that the title that I picked for this is a little bit hyperbolic because, again, he does correctly recognize a bunch of things. He gets close. But then at the end, you'll see it's not actually hyperbolic at all. The very end of this,
Yeah.
like the last little bit that we're going to talk about, oh, my friends, you all are going to lose it when you see the part that caused me to lose it when I went through this when I was taking notes. I went, hang,
Yeah,
no,
yeah.
but
It
he
gets
did.
pretty
He
wild.
did. Yeah, it gets wild. The end is it goes off the rails in Act 3.
Yeah,
So,
I think a lot of creationists are like this, and that they know just enough terminology, they've read just enough of the textbooks to think they understand it, to think they've got it down, and can like think they're formulating these novel thoughts, but they've not quite read enough. Like they're not quite there to really
close.
grasp the concepts, and so they just miss it hard.
And you see that my favorite example of that is Jeanson in his famous debate with Dr. Herman Mays when he quotes the genetics textbook, the Fatuma textbook, get mispronouncing the man's name first of all because he
Yeah.
never had any formal instruction on any of it, right? He just
Yeah.
kind of, winging it. But then
Yeah.
he's like, here's the equation, and God bless him, you've got Herman Mays sitting there going, that's like the first day of class.
Yeah.
And then there's complexity on top of that. Do you realize that there's other things go? And Jeanson's just like, nope, that's the equation. We just plug the numbers in and that's it. We're done. Right. And yeah, so I think that is a
Because they
very
don't have to
astute
read any farther
observation.
than like intro textbooks, right? Like they don't
Yeah.
actually like follow the field anything beyond what you would read in like an introduction to the class.
Yeah. Also, this is got $5. Thank you from Little Pink Bowser. Broke the clickbait slash headline question rule. The answer should be no. Yeah. When did Eve live? Can we know from science? Yeah, that's the headline rule. Okay. There is. Yep. Thank you. Betteridge's Law. Thank you. I'm glad somebody knows what we're talking about here. Okay, so let me pull up the video, and we're not going to start it right from the beginning. We don't need to get the intro stuff about this lovely park that Dr. Carter is walking through. We're going to jump to Zach. I think you had the first timestamp at about 4.5 minutes is where we want to land.
Yeah, yeah, right around there.
All right, so pardon Dr. Carter's going to jump right in, folks, and then I'll move him along.
Rob, welcome to Biblical Genetics. Hey, if you
All right.
don't agree, especially because I don't agree that you
Oh,
can use
hang on now.
an African branch that's outside all of the other branches.
Do you want me to go back a little further?
I can also just like explain what he's trying to show here.
Yeah, go for it.
Yeah, so basically, he's trying to make this argument that this was the first phylogenetic tree of humans based on the mitochondria. I think, what did he say? This is 87. Yeah, he's got it here. So this is like the first phylogenetic tree that was built, and he makes this claim that the reason that they think that the human mitochondrial Eve is in Africa is because of this branch that he circled here. And there are these long branches, and he doesn't agree that mitochondrial Eve is actually from Africa because he rejects the molecular clock. That's what he's going to go on to say here. And the reason that I want to bring this up is because this is like a preposterous argument. We're not the idea that mitochondrial Eve is in Africa is not based on the length of any branch. You can actually have like really long internal branches that are being driven by things like selection, right? So like positive selection increases the length of branches because you get rapid fixation, right? And that can happen internally. What the reason that we say that the African lineage represents the ancestral lineage or that mitochondrial Eve occurred in Africa is due to the pattern of clustering. And so all of the out of African populations, the European and Asian populations, are nestled within a broader phylogeny that includes African populations, right? So you do have a longer one that represents like the Sand people and various other ones, but nestled within that are other more closely related African populations that are more closely related to Europeans and Asians than they are to other Africans. Now you cannot get that pattern in any other way except for an ancestor of that African lineage split off into Europe and Asia. That's the only way to get that nested pattern. And so it has nothing to do with the molecular clock. It has nothing to do with the lengths of the branches. It's about the shared patterns of ancestry that nestle Europeans and Asians within that phylogeny.
And you can
That's
see
the
that
main thing
right here. And you can see that right here on screen because the solid circles represent African lineages, and you can see we have in the bottom right, we have the kind of that longer branch that Carter points out. But then you can look throughout the rest of the tree, for example, by the number 40 near the top right, or up by the number 70 or 80, or over on the left, just below 110, you have other African branches, which is exactly the predicted pattern if the common ancestor of everybody on this tree was an African population, and then everyone developed from there.
Yeah, I've did a video several years ago where I took a bunch of mitochondrial genomes, a bunch of human mitochondrial genomes, and built a phylogeny to basically show that. It was a lot more than shown here, and you can clearly see that the European and Asians nestle within that broader African family tree. So yeah, again, he just completely misses the reason why we say that mitochondrial Eve is from Africa has nothing to do with the molecular clock.
So now, besides 650, because in the span of like 20 seconds here, there's some issues that I think we're both going to want to talk about. There we go. Let's just drop it here.
Problem fixing mistakes. That's why we get mutations. All right, stupid illustration, but just popped in my brain. So one way that they explained this single human female ancestor, which was a bit of a shock, was not predicted. It was folded into evolutionary theory by invoking a bottleneck. Now, the bottleneck has other things later on that they also use.
Okay. Let's just pause on that. Like 15 seconds, there's two really big problems there. First is the idea that a single mitochondrial most recent common ancestor was a surprise. Zach, you're far more well-versed in population genetics than I am. But my understanding of how this works is that such a most recent common ancestor is not only not a surprise, but it is a mathematical necessity as evolution happens.
Yeah. And it's a mathematical necessity that goes back to the 1930s. Basically, when you get back to like 1931, Wright first worked this stuff out. The people don't often realize that it's so old, but it's because Wright worked forward in time, and often we think about things backward in time. So we often think about the coalescent, which wasn't developed until the 1980s, but really the coalescent is the backward version of forward time Wright-Fisher genetics. And so really the way to think about this is if there were ever more than a single common ancestor or a single ancestor, what that would mean is that there was some original population that just had things created in place. Right? That's the only way to have multiple ancestors for any given population. It means that population has no common ancestry. And that's exactly what evolution says did not happen. In fact, creationism would predict that there would be multiple ancestors or multiple lineages that don't have ancestry. Evolution requires there to only be one ancestor. And you can think about this as, imagine a mutation emerges in a population and just one individual has that mutation. And then over time, that mutation eventually spreads to everyone in the population and goes to fixation. Now, everyone at this end of the population that has that mutation has one ancestor, and it's that individual where that mutation arose. Right? That's the idea. And so long as drift happens, right? So long as a mutation can go to fixation, which is like a basic prediction of evolutionary theory and a basic prediction of just, genetics and population genetics, then you will only have one ancestor. And that's true across the entire genome. For every little genetic position across your both nuclear and mitochondrial genomes, there is just one ancestor, right, that you can trace back into time. And so the idea that we predicted that there would be multiple mitochondrial lineages is just preposterous. It's mathematically impossible.
Yeah. And then the second thing he says there about how, well, to explain this, we invoke a bottleneck. And this is something that creationists, I really think this comes down to that god-awful why should mitochondria define a species paper that creationists love so much. I think it really comes down to creationists just glomming onto that paper and most common ancestor equals bottleneck. It doesn't. It just doesn't. Like, I don't know what else to say when they're like, oh, and the most recent common ancestor indicates a bottleneck. No, it just doesn't.
Carter tries to make this argument that the only reason that like evolutionists claimed that there had to be a bottleneck to explain there only being a single ancestor, right? Like that's what we are invoking to explain that. And again, that's preposterous. It doesn't matter where in the genome you look. So if you look at the most recent common ancestor in the nuclear genome, it's much, much further back in time,
right?
Because there's a lot more diversity. It takes a lot longer to get back to that original ancestor. And so it doesn't, we're not trying to invoke bottlenecks. The bottleneck was inferred based on current patterns of genetic diversity. So when we looked at the genome, we saw there's a lot less genetic diversity relative to how many humans there are on the planet. What that implies is that there must have been a much fewer people in the past. Now, importantly, it's not just about standing levels of genetic diversity. It's also about the shape of the site frequency spectrum. Never heard a creationist talk about this, but it's the clearest indication of a bottleneck. Not genetic diversity. Genetic diversity is a little bit more difficult to interpret. The site frequency spectrum is not. So in a really, really quick, simple way, the site frequency spectrum is on the x-axis. You count up the number of individuals that have a given allele. So at one end are individuals that only one person has it. Those are called singletons. If two people have it, they're called doubletons, etc. And so what you expect is this sort of negative exponential going down like this. Most of the mutations are from singletons, and then you get fewer and fewer and fewer and fewer and fewer, right? Now, when you have had a bottleneck, you actually find that there's a little bump out here. And the reason that there's a little bump is that a lot of those mutations that were at really low frequency when the population was large, when you shrink it, they suddenly jump up in frequency because now very few individuals, a lot of them now have that mutation and it rises to high frequency. When you look at the human site frequency spectrum, you see that little bump. And that's a clear indication that we must have gone through a very recent population bottleneck. And so again, you don't have to just rely on genetic diversity. It's also the shape of the site frequency spectrum that allows you to infer both when it happened and how severe it was.
I love it. I did not expect us to talk about the site frequency spectrum here.
You brought a population geneticist on, you know you're going to hear about it.
I love it. I love it. I also, just before, I don't want to lose it, I don't want to forget, we got from Tad is Mad for $2, crime number 2, not in a boat. I won't forgive this. Dr. Carter is somewhat famous in, our circles for the locales of his videos, which I love personally. It's always like, where are we and how does it relate to what we're talking about? And he does a really fun one with that at the end of this video. He really uses the location effectively in terms of what he's talking about. But he's got some really good ones. The famous canoe video is a kayak. I forget which, but there's one where he's in a boat, and that one is very popular.
Carter is always fun in that he's always starts it off with like, I am in this wild place all of a sudden today, and here's this thing behind me, and you gotta love that about Carter.
My favorite one is the one where he was literally in a cave, like in water. That was my favorite one. It was
that's good. I haven't seen that one.
Oh, it was very dark. Like he's literally in a cave. Yeah.
Nice.
Okay. Let us continue. I'm going to jump ahead a bit. There's a bunch of stuff in here that I want to point out that we don't need to watch the segments because we'll be here all night. But at one point he says, we didn't know, talking about similarity to other apes, we didn't know if chimpanzees were the ancestor, and it's we never thought chimpanzees were the ancestor. It was a question of who was the sister group, which is not an ancestor, whatever. There was that. He did say that, in fact, this one I'm actually going to play up at 10:30 about the 3 nodes thing. Now, this is a little bit of inside baseball, but this is stuff that I've hammered creationists on this over the years, so I like that Rob Carter is dropping it. Where they used to do this thing where they'd be like, look, there are 3 nodes in the mitochondrial phylogenetic tree, and those represent Noah's sons' wives, and that's whatever. And he doesn't do that anymore, apparently. And let's just watch that little bit because it's I think, notable that he has changed that specific thing that he used to say.
Different mitochondria from Eve, or 2 mutations could have picked up in that 1600 years between Eve and the flood. There could have been up to 3 different mitochondrial lines. But I no longer say there was 3 lines. I said that earlier in my career, my creationist career. But no, because What if two of the girls were sisters? Well, then they would have the same mitochondria, and then there would only be two mitochondria. What if all three of them were sisters? Well, then there's only one mitochondria. Mitochondrial Eve was their mother.
What if they were
All of the
triplets?
mutations.
Right. Like, so that's a good example of where he's like, you got it, and we're over here.
And we lost it.
Yeah. Yeah. So I just wanted to point that out because this is a thing that like creationists have used. I think it's a figure that he made actually for the longest time. Creationists be like, look, there are 3 nodes, just like we predicted in the mitochondrial thing. And he's like, no, I don't do that anymore. Which like, hey, look, progress, right?
Yeah.
Progress. At least
Yeah,
we are we're not doing a Donnie Deals fallacy. We're not just saying the same thing, which is good. We like that.
It is nice to see growth. It's a little unfortunate that growth then kind of like grows out of control and ends up like doing even more like Genesis fan fiction stuff where it's like, well, maybe they're sisters, maybe they're cousins, maybe God just created a bunch of different mitochondria and put it in each. Like, it could be anything, you know.
Which is where it always ends up, but we'll get there. So let's go now. This is where we get to like the actual kind of crux of what we want to talk about here. So we'll go to 11:30, and we're going to do this for about 20 seconds. And Zach, if there's any time points in here that I'm skipping that you want to talk about, let me know. We'll do about 11:30 to 11:50, and then we're going to jump to 13 something.
Okay.
But if there's anything in there, let me know, and we can catch that too. Here we go.
Just a little funny little anecdote there also.
All right, not bad.
But because both sides can accept the data, therefore the timing is critical. The timing of Eve is 100% the argument. How do we put a date on Eve, or if you like?
Yes, that is like the whole point, because the whole point of this years-long conversation we've been having with Dr. Carter is mitochondrial Eve didn't exist 6,000 years ago, right? We can do the population genetics, calculate the substitution rates, we can do the phylogenetics. Mitochondrial Eve did not exist 6,000 years ago, and creationists are always like, no, you take the single generation pedigree rates, you extrapolate those back, boom, 6,000 years. That's been the whole thing. So yes, that little 20-second bit is the setup for everything else we're going to talk about here, right? That's what we're doing. So now we'll jump to 13. I got 1345 next, but Zach, if there's anything in there you want to hit, let me know.
No, I think we can go there. I mean, the only things that I had was that he talks a little bit at 10 about how there could be any number of mitochondria within the eggs, and so mitochondrial Eve could have many lineages. And I was this just makes an unfalsifiable claim because you could basically make anything fit the data, but he's going to meet many of those throughout anyway.
He's been doing that for years. There was the Patriarchal Drive paper that he did, or it was a conference. It might have been like a conference report. I forget if it was in the proceedings of a conference or if it was in like one of the actual, not like CMI's journal, not Journal of Creation, but it was like Creation Research Quarterly or something. It was like actually like a Patriarchal Drive paper. I think it was him and Sanford and somebody else. And one of the things they talk about, I may be misremembering it. I apologize to everybody involved if I'm misremembering the source, but one of the things they entertain in this thing that I'm remembering is the idea that you could have created germline diversity. And this was, that's what it was. It was a solution to it was basically like a created heterozygosity thing. And it was like, and also, you could get more with germline diversity. I'm not saying that was the case, but it could have been.
Yeah.
And so he like entertains that notion fairly regularly over, the years of this conversation.
Yeah, and it's nice because when you can just make up anything and you can just invoke magic to explain anything, then you have any model you want. You can explain any bit of the data that you want. And it's these like consistent, unfalsifiable Genesis fanfic stuff that just drives me crazy about creationists.
It would have made like my dissertation defense so much easier. But why is it that way? Because I imagined it that way.
Yeah, because wouldn't that be fun?
Wouldn't it be neat if it worked that way?
Yeah. Gosh,
Yeah.
that would have made my PhD so much easier.
So before I lose track of it, Tad is Mad is back for another $2. Thank you, Tad. You don't have to keep doing this. Crime number 3, bringing magic to a science fight. That should be illegal. It's not allowed. Magic to a science fight. All right. So in this bit, this is very short, but it's important because I have saved this. I've bookmarked and timestamped this little bit, and you all will see why in a second. This part I like.
That's where the African Eve idea comes from. Now, because I reject a molecular clock,
What? You reject the concept of a molecular clock? You should talk to Dr. Nathaniel Jeanson, who bases his entire career on the strictest molecular clock anybody has ever encountered in their lives.
Yeah. It's also kind of funny because he's going to, I totally missed this because he's going to go on and make the argument that the mutation rate equals the fixation rate and like not that much longer. And that only works. And that's
a molecular clock
argument for a molecular clock, right? So if the mutation rate equals the substitution rate, then the molecular clock is very easy to demonstrate. Now, a molecular clock doesn't have to work that way, doesn't have to tick because of genetic drift, but like, if genetic drift is the explanation for the diversity, then that by necessity must be a
That's how it works.
Molecular clock, yeah.
Yeah, that line jumped out at me as notable, and I will be, believe me, folks, I will be pointing out that, well, Dr. Rob Carter says there's no molecular clock, so on what basis is Jeanson doing anything? Jeanson needs to defend his work conceptually before he defends his methodology specifically.
Yeah. Yeah.
And now we just have the creationists fighting with each other over whether there's a molecular clock or not. And I'm just and I'm just sitting here with my popcorn.
Yeah.
Chat. Anytime someone brings up Jeanson, you can now throw Dr. Rob Carter at them and say, but Carter says there's no molecular clock. What's Jeanson's response to that? Yeah. Thank you, Rob. Thank you for that moment. All right. So now we are going to jump. My next timestamp is 15:15. And now we're getting into some nuts and bolts here, and it's, I think, notable, some of the things that he agrees with us on.
Yeah, I'll just say really, really quickly about just some of the molecular clock stuff that he's going to say here. He goes on to talk about how he thinks that there's a lot of like heterogeneity in mutation rates across people and stuff like that. And I just want to say, like, we know, like we know this. Every phylogeneticist knows this and has for a really long time. I mean, going back to like, again, Tamurin and Ney came up with like some of the substitution models that allow you to adjust the rates for transitions and transversions. Those are sort of the first models. Now we have GTR models, the general time reversible, where you have like independent rates across every kind of transition and transversion imaginable. And you can add like gamma parameters to them. So there's like, we can very easily model rate heterogeneity. It's something that we do with every phylogeny we make. No phylogeny is built assuming a strict molecular clock. It's just not done that way anymore.
When I was in graduate school, a couple of my colleagues in my lab, and we did viral evolution, viral phylogenetics was our thing. And one of the things we did was like empirically determine mutation spectra and base specific mutation rates. And so one of the things that a couple of my colleagues developed specifically for use in viral phylogenetics was what they call the unrestricted substitution model, where it's GTR, general time reversible, but not reversible. So each rate was independent, right? So instead of A to T and T to A being the same, no, those are now independent rates you can model. So, right, like, nobody thinks this. Yeah. Nobody thinks that this is... Yeah.
And we even have things like we will vary the rate across even different sites. I mean, like the program partition finder is old at this point. It's like kind of out of date at this point. But like
region specific, you can have region specific mutation rates.
Yeah, we would partition out the sequence, and then you would re-estimate the substitution rates across each of the individual nucleotides, right? So you could even
Yeah.
get it to the nucleotide level and capture the rate heterogeneity. So again, like they're talking about as if the field has been in the late 1980s, early 1990s, and has like not changed in 40 years. It's just like it's kind of wild to me.
Right. Another point to this that again a colleague did when I was in grad school is a really cool thing. She was looking at viral substitution rates as a product of the rate of cell division of the tissue they infect.
Oh, that's cool.
So looking at are the faster dividing tissues, are the viruses having to adapt to the shorter generation time of the host cells? Because you probably don't want to be replicating yourself when the host flips into mitosis mode, right? So that's
Yeah.
going to impose a shorter replication cycle on the virus, which means more base replications per unit time. So do you have an elevated rate in a tissue specific way based on which tissues you infect? And
that's
she
really
did
cool.
and she did find heterogeneity across different tissues. So she's looking at like muscle tissue versus epithelial tissue and like
Yeah.
the answer was yeah, there was the faster dividing tissues tended to
Yeah.
have viruses that had higher substitution rates
Oh, that's
because
super
the
cool.
yeah, right. So
Yeah.
this is one of a million examples of a thing that people who are actually practicing in the field are like, yeah, why is this a thing? And the creationists are like, shocking development,
Yeah. Yeah.
right?
Yeah. And it's like really it's kind of intro to phylogenetic stuff. When I teach phylogenetics and I go through sort of the basics of like phylogenetic methodology, we talk about substitution models, and you inevitably talk about rate heterogeneity when you talk about
Yeah,
substitution models.
you can't just assume everything is the same. Shocking. Absolutely shocking. I will also point out that creationists know this, just not when they're talking about this. But in other contexts, they know this. Because ask them about things like mutation hotspots
Yeah.
and the implications for like directed mutations, right? When you have mutation hotspots. And ask them about species that can artificially increase their mutation rate in response to some environmental stress, right? So creationists know this outside of this context when it's inconvenient.
It's they're able to like move the parts of their knowledge around to make sure that none of the pieces ever come together in such a way that would like lead to extreme cognitive dissonance.
Wait
you know?
a minute.
Yep, that they have to be sure that these pieces are isolated from these pieces. That's why it's like they know all the stuff. All the knowledge is there. It just won't come together, you know?
Yeah, this is a silly analogy, but I'm reading a book right now. I'm actually rereading a book right now because it's a really fun book and I read it a bunch of years ago, so I'm going back to it. It's called Atomic Accidents. It's about like the history of basically research and energy and industry and military uses for all atomic stuff. And one of the recurring themes is criticality accidents, where you accidentally achieve a critical configuration where you have like uranium dissolved in a solvent, but you put it instead of in a tall cylinder, it's in like a tomato can shaped container. So it achieves criticality briefly.
Yeah.
And it's like what I see going on in, not like Rob is the example here, but they have the knowledge in a non-critical configuration, so it never achieves criticality.
Yeah, that was a fun walk.
You're welcome, everybody. That's why you
Yeah.
come here. All right. So in this section, we're going to talk about mutation rates and long-term accumulation rates, which the technical term we want to use is substitution rates. And let's just see what Rob has to say here.
I know it, but I do know what's going on, at least in that respect. So here's the deal. We've got a mutation rate we can measure, and we have a long-term accumulation rate, and they're not the same thing. The long-term rate should be a little bit lower than the rate we can measure for multiple reasons. But
Okay. I just want to pause there and say, broadly speaking, yes, substitution rate is going to be lower than mutation rate. Little bit lower is like, it's a little, you're shading it a little bit, but like for creationists, that was really good.
Yeah.
That was real for creationists. That was really good. And then we're going to talk about a bunch of reasons why this might be the case. So there's like a minute and change of kind of stuff here, and then we could talk about that. And then there's a little bit more before we get to like, here are the 6 reasons. And that's where a lot of what we want to talk about is. Okay. So, Zach, do you have any thoughts before we continue this next, like, it's a little over a minute. I got 15:30 to 16:45 is our next
No, I'm good. Let's do it.
stroll. Let's do it. Let's hear from Rob. This part, all the parts that I'm playing, I think are important and interesting, but we're getting into the meat of it now.
When we measure it, when we look at family trees, we know how many generations or how much time has separated 2 people. We get a whole range of measurements. The earlier ones were really high. Earlier ones were just on the control region, was like 1 to 1.5 mutations per generation. Woo! Those haven't held up very well.
Tell that to your colleagues, sir, who still cite Parsons' 1997 as their go-to mitochondrial mutation rate paper.
Yeah, a lot of those early ones we found now that there were like somatic mutations. There were like a lot of things going on, and we know actually that a lot of the mitochondria in the soma are under much weaker selection, and so tend to actually accumulate mutations much more rapidly in somatic cells than they do in the germline. So you would expect high rates of mutation, especially in the somatic cells.
And we're gonna mention that later, because he says like, oh, we don't have to take care of this, but then some of the studies that creationists love to lean on explicitly don't account for that problem.
Yeah.
In their data, like you can ask the authors, and someone did in one case, and the answer is nah, we didn't account for that. So like, we'll get there. We'll come back to that point a little bit later on. So that's yeah, those early studies are not great. Even some of the more recent ones, same problem.
Yeah.
And now that we have full genome sequencing and millions upon millions of mitochondria that are available to DNA testing companies and things like that, and a lot of family studies have been done with known family trees, our mutation rate, 0.03. That's 1 mutation every 30 generations. And it ranges up to, I found, up to 1.5, but most of the modern ones are about, 0.05, 0.7, things like that. So it's less than 1 mutation per generation, which is fine for now. Just hold on to that. That does mean that Eve didn't live 6,000 years ago. Those slow rates are too slow for me, but they're still too fast.
All right, hang on. We'll get to that sentence. We'll get to that next sentence in a second. But I just want to point out that Zach, we are making progress in this conversation, I feel like, because that would not have happened 5 years ago.
I don't know another creationist I've ever heard say that, right? Like ever say Eve couldn't have lived 6,000 years ago based on the mutation rate. Like I've not heard another creationist say that before. So yeah, no, I'll take it. I'll take the W there.
It's why we appreciate Rob. The scenic views, the on location reporting, and like acknowledging basic facts about population genetics related things is nice.
Yeah.
So now let's get to the next part, which we're gonna go for about another like 20-ish seconds. I think I get 20 or 25 seconds there.
Slow for me, but they're still too fast for the evolutionist. That's really funny. None of us like the number. Eve should have lived less time ago than those really low mutation rates would indicate. But for the evolutionist, if you take those low mutation rates, then Eve was not 200,000 years ago. She's less than that.
We got it.
I don't. It's very funny to me that he thinks that like I'm just going to use his term. Evolutionists were ever bothered by the mutation rate, as if we knew or like that we expected that the mutation rates and the phylogenetic or substitution rates would be exactly the same thing.
Yeah.
Like that's just not a prediction, and like it's not even a prediction if you go back to Kimura, right? Like Kimura was about the neutral mutation rate. This is what they don't seem to they always like to drop the neutral part and they just say the mutation rate. but the mutation rate is the totality of the rate, which includes deleterious mutations, near neutral mutations, and benefit. It includes the full spectrum.
Everything.
The full spectrum, the fixation rate will not match the mutation rate of the full spectrum, and the theory never predicted it would. It's only those that are strictly neutral that those things should match each other. So it's very puzzling to me that he's like, oh, the evolutionists, they were so mad and so upset about this number, as if no, we never cared. We knew those numbers were going to be different.
Yeah. And also, let's just appreciate, so we've got Zach here to give us the history and the population genetics of it, and I'm here to point out the heinous nonsense that he's pulling here. I want everyone to see the bait and switch here, because this is where I get off the Rob Carter train, because oh, it's way less than they thought. How much less, Rob? The answer is that 200,000 years is the high end of the estimated range, and the low end that you will find is 60,000 years, aka 10 times longer than the young Earth timeline. Mm-hmm. So, an evolutionist, when I describe this, I say, 100 to 200,000 years, some estimates are as low as 60,000 years. And if we're having a longer conversation, I will often follow that up with, but when you put that together with other evidence, like the archaeological evidence of the timing of the out of Africa migrations, those migrations were probably earlier than that. So the low end of that range, which again, you're calculating this and then you get a margin of error, you get a range, the low end of that range is not consistent with other evidence that we have, whereas the rest of the range is consistent with those other lines of evidence. And it's like you said at the top, Zach, it's a video, there's less nuance here, we're not going to get that level of caveat and kind of checking yourself, but also he doesn't do that in the article either. So
yeah. I think it's important to note that we don't just measure the rates of change just based on genes. We use external sources as well. We use biogeographic data. We use archaeological data. We can use paleontological data. We use lots of other sources of data to help us check our molecular rates. And so we are constantly cross-checking these things. And that's why they tend to be pretty robust. And that's why over time, our confidence intervals have narrowed and narrowed and narrowed, because we get more and more data. We get more and more genes. I don't know why we're still talking about mitochondria in 2025. We sequence the whole genome now, right?
We have the whole genome.
We've tracked the movements of the entire nuclear genome. And so the most recent papers, there's been some just incredible papers out in the past couple of years tracking the migration patterns of humans across the whole globe by looking at not just the entire extant people groups, nuclear genomes, but we've got people that were alive 3,000 years ago, 5,000 years ago, 20,000 years ago, and we can incorporate those into that migration pattern as well. The new stuff is so incredible, and we're out here still talking about mitochondria stuff like it's in the 1990s. Creationists were always stuck in the past, man.
I mean, that's par for the course with creationists. We're always going to be 30 years out of date here. The other thing with that, though, is we talk about these cross-checking and everything, and the word that creationists love to hate with that is calibration, because when you calibrate your mitochondrial substitution rate against known migration events, we know this island was settled at this time, therefore given the two populations, we can put a marker in a hard spot in the past and say the curve must hit that spot, right? And you're manipulating the data, you're ignoring the data and posing your own evolutionary lens on it. No, these are things that happened within the last 3,000 years that we know happened because people were there and we kept track of it.
Yeah, it's also just basic Bayesian statistics, right?
Yeah.
The idea of a good Bayesian knows that you should bring in all prior knowledge with you to update the model, and so that's why we're called priors. No. And so if we have like if we have these dated time periods, we can put prior distributions on them. We can include the mutation rates as well. Like we can include the genealogical rates as a prior. We include the migration rates as a prior. We include other archaeological evidences as a prior. And all of those things interact with the posterior, right? And so we get a holistic view of what that date should be. And it's consistently over 100,000 years when you include all of that evidence together as in part of your model.
It just sounds like a lot of evolutionary assumptions to me, Zach. I don't know.
Statistics is an evolutionary assumption to them.
Right. Recorded human history is an
Yes.
evolutionary assumption. All right, so we're going to pick this up here. There's a bunch of stuff that's just like not relevant. So we're going to jump now to about 1850. Because this is where we're getting into, like, there are 6 reasons that the substitution rate and the mutation rate would diverge. And that would cause mitochondrial Eve to be further in the past than you would expect from the single generation mutation rate. The first one we're going to talk about is selection, and that's like the big one, and he spends like a bunch of minutes on that. Mm-hmm. And then all the rest are very small, and we can handle those very quickly. But the selection one has a bunch of stuff we're going to want to talk about, because this is the main thing. The main contention here is that selection plays a very minimal role in shaping the sequence diversity of the mitochondrial DNA. It plays a very small role, insufficient to get you from your single base mutation rate to the evolutionary substitution rate. Selection can't get you there. So let's break this down.
Mutation rate. But here's my 6 points. Point number 1, selection. This is Creationist big argument. He basically said that this proves Carter wrong, whatever he actually said. It's been a long time since I watched that video, but
It does. To be clear, it does. The fact that there's strong selection operating on the mitochondrial genome does prove him wrong. Just not to bury the lead, like, it does. Straight up.
he thinks that this proves me wrong. And yet, there's multiple problems with this. First of all, mathematically, I'm the one who ran the calculations. I ran a lot of simulations to try to figure out the difference between the mutation rate and the accumulation rate using a population model. To my own population software indicates that, yeah, you can remove some, but not many. You can only remove a fraction without killing off your population, or basically most mutations are nearly neutral. Everyone believes that. Therefore, you can't remove most mutations. If you wanted to remove a mutation, it has to be selectable. It has to rise above the noise, the environmental noise, the background genetics noise. It has to have an effect on the individual. You, right now...
Okay.
Oof, there's a lot there.
There's so much there. The hard selection thing, we're just ignoring soft select, with just there's...
Oof, we opened straight off the bat with Mendel's accountant, actually. So like that
so like,
paper,
let's start with that.
That paper that he showed, I think we've actually talked about this paper before on a stream. Maybe I'm misremembering, but that is done with Mendel's accountant. And for folks that don't know, Mendel's accountant has been demonstrated to be a rigged program. There's a really great Reddit post. This was maybe 6 months ago. There was I forget the user's name, but he did a little bit of digging, and him and I had like a back and forth where we did some calculations. And it turned out that Mendel's accountant has this, they call it a noise scaling factor. And basically, it's this little variable that they tack on anytime that you have a beneficial mutation, and it downweights that beneficial mutation. And so, in effect, for the exact same selection coefficient for a deleterious and a beneficial mutation, you're just swapping the signs, the beneficial will always still be less than what the deleterious impact will be based on the scaling factor that is default behavior in Mendel's accountant. And when you look at Carter's stuff, he says, I keep the defaults in Mendel's accountant, which is why his simulations look like they do, because they're artificially deflating the selection coefficient for any beneficial mutation. And so all the populations go extinct because they can't, there's literally, it's impossible because there can be no compensation from any beneficial mutation. It's just
they're cheating. They're cheating. It's like, yeah, they're cheating.
So yeah, so like that whole the entire paper and
Forget that.
its simulations is based on a rigged program. Do we want to talk about the nearly neutral stuff?
I just want to, it's a real quick thing, but he slips it in there. He does this thing that a lot of creationists do where they talk about, I think Sanford's done a lot of this, where they talk about how basically if you're going to have effective selection, your population would go extinct because you have to kill so many individuals to get rid of the harmful mutations. And I know you've hammered this point so many times, and here we are in the year of our Lord 2025, and you've got a young earth creationist still acting as though hard selection is the only type of selection that exists, where either you're fine or you're dead. And that's not how it works. You could have an average of 0.8 kids instead of 1.2 kids. That's still selection against whatever it is that you have, right? So, like the whole thing about, well, in order to remove all these mutations, you'd have to kill all the population. No, you don't. Just, no, you don't.
Yeah.
It's more complex than that. Just,
Yeah,
we've said this, both of us have said this multiple times.
Yeah. It's like they misunderstand that. So like selection is really only hard when a population is adapting to a new environment and they're having to sort of climb that peak, and you're actually having more and more children, right? So you're adapting, you're growing in size. Like that's when selection tends to be hard. But for most of a sort of species life, they're sitting on an adaptive peak. Adaptation is pretty rapid. And so they're sitting on that peak, and now the way selection is acting is competition between individuals, right? So it's not about who's having more offspring, it's about who is able to get resources better and outcompete their neighbor. And in doing so, it's like, I don't have to outrun the tiger, I just have to outrun you, right? Like it's that kind of thing. And that's a very different kind of selection than the kind of selection that's on like reproductive birth rates, right? And those two different ways of like dealing with selection have really big impacts on how purifying selection can remove mutations. In hard selection, the genetic load can get too big because you can like not have enough children to keep up with the genetic load. But when you're talking about soft selection on top of an adaptive peak, the genetic load is almost impossible to get high enough to drag it down in soft selection because it's just so small. And in fact, it's basically just like the genetic variance is the amount of the load, and the genetic variance is always really small, right? Like the genetic variance in humans is less than 1%, right? And so like it's just impossible for soft selection to ever generate a strong enough genetic load to drive the population extinct.
I want to just pause because we have another $2 from Tad is Mad. Crime number 5, absolutism. Oh, and did I miss the other one? I think I missed one actually. Crime number 4, our data are the same except when not. Which, yeah, a lot of creationists do that, and we should coin a term for that. I mean, a lot of them do the same. It's, same data, different interpretation thing, but there's the data are the same, and then 5 minutes later they're like, these data are different.
Yeah.
Okay, so now we can get to the nearly neutral part of that. Zach, I feel like you're a really good person to talk about this because this runs up. He's walking right up to the line without saying the words genetic entropy, and I feel like you have thoughts on that topic.
Yeah,
and maybe even wrote a paper on it.
It's I know Carter has seen our paper by the time he made this video.
Yeah.
And I know that because he emailed me and asked for a copy of it.
So
I know that Carter has seen our paper, which makes me it hurts me a little bit that he gives this whole speech about near neutrality and how selection wouldn't be able to work and all this when he's like, we did the math on this. We did this paper that shows the exact opposite of what he's claiming using realistic distribution of fitness effects. And so it's a little frustrating to me. Again, I'm doing the like butthurt, like they didn't call me back kind of thing, because it's like I thought we had like a good rapport, and then it turns out you actually didn't read the paper, and that that kind of hurts my feelings, Carter.
Yeah, he emailed for the paper.
Yeah,
and then
he never called me back. Yeah,
either he didn't read the paper, he read the paper and didn't understand it, or he read the paper, understood it, and he's lying about it. Those are our three options here.
Yeah.
Let's just go with he's ghosting you is the most generous interpretation.
I think so. I think I've just been I've been ghosted by Carter, and you know what? That's okay. It's all right, Carter. I will just for the context for people, the way that the paper basically worked is I looked up a bunch of like distribution of fitness effects and a bunch of different papers where they had measured it empirically. If you don't know, distribution of fitness effects is on the x-axis. You have what that mutation does to like either growth rates or reproductive success or whatever way you're measuring fitness. In the middle is the zero, that's strictly neutral. To the left of it into the negative territory or deleterious. To the right of it are beneficial. And then there's some distribution of mutations de novo mutations that happen every generation. So we went through, we found a bunch of distribution of fitness effects, and then we kind of modeled our simulation based on those distributions. So we tried to get like a realistic distribution. And we were also trying to match what Sanford had done before. So we wanted it to look close enough to Sanford's that they couldn't be like, well, you just totally made it up. But we also wanted it to be realistic. So we ended up doing like 99% of all mutations are strictly deleterious. Whereas only 1% of them were beneficial. And I limited where that beneficial could be to all less than 1%. So they were like really, really, really tiny benefits when they were beneficial. And so we ran that simulation to see if that population would go extinct, as Carter is here basically claiming that they must go extinct, right? Selection, there's way too many deleterious mutations. And of course, and like, again, population genetic theory already knew this. I don't think we showed anything that was really novel. This was already known. We just put it in the way that I hope creationists would see and deal with. But in effect, we showed that the population won't go extinct. And the reason that it doesn't go extinct is because those beneficial mutations, while there are so much fewer, when they do occur, they fix very rapidly, right? And so they can quickly overcome the deleterious ones that can only fix by drift, right? So most of the times they're going to be lost to drift, or they're going to be lost because selection is driving them away, right? And so those two things keep the deleterious rate lower, whereas the beneficial rate, even if it's really, really small, can completely compensate for it by just a little bit of selection rapidly fixing them. And we showed that. We showed that with 5 different replicates in that simulation. So I don't know. And Carter, I know Carter has that paper. He knows that what he's saying isn't true. Yeah. I assume. Yeah.
Have any prominent creationists ever responded to that paper? No.
No, not even unprominent creationists.
Yeah, no, not even YouTube randos will not touch that thing.
I managed to talk to Donnie about it for like 5 minutes when I was having the Jerry Bergman debate, because Bergman was like, I don't know, falling asleep or something. And so like me and Donnie talked for a few minutes about it. And that was like the closest I ever got to actually talking to a creationist about that paper. So,
yeah. Yeah. All right. So there was a lot there in that about a minute that was wrong. And again, the point that Dr. Carter is trying to make here is that selection is not going to slow down the substitution rate that much from the mutation rate. And he's going to continue to be wrong about this in other and more creative ways as we continue. So I'm going to jump just a little bit, just like 20 seconds here that don't matter. I'm just going to jump a little bit. Here we go.
Actually selected away? Probably not. In fact, yeah, there is a whole lot of selection in the mitochondrial genome. I looked at all these lined up sequences and I calculated about half of the mitochondrial genome is invariant. And that can mean one of two things. Either it's not very old. There's a truck coming up the road here. Either it's not very old and we haven't had a long time for mutations to accumulate, or more likely, there are a lot of mitochondrial mutations. Does this guy go by?
I think you're muted, Dan.
Let me just take a minute here to link the paper in the chat as requested. And if you can't access the full text, you can email either me or Zach. We will happily send it to you. There's a little intermission here where this little cart goes by with the maintenance guys. But he gets back on topic just real quick right here.
There are a lot of mitochondrial mutations that are deadly, that are instantly removed from the population. Yeah, that's definitely true.
Okay. Now that, okay, so he's now talking about like hard selection, but specifically for the mitochondria. And this is important because a lot of the selection for mitochondria happens in the germline, where if you have a harmful mutation among all those germline cells, the ones that have the mutation will not propagate. They will not develop. They will die, right? So that is an important thing that's going on there. He has identified a very strong source of selection in mitochondria.
Yeah, and this is like broadly speaking called gametic selection, right? So you're selecting between gametes. Yeah.
Why does now? I just want to take a minute to put this together with the last thing he said about how you get a bunch of mutations that are neutral, nearly neutral. You can't select those mutations, right? They are below the selection threshold. Okay, Rob, let's take a minute and think about this in the context of the mitochondrial genome. The mitochondrial genome, as creationists will always remind us, is uniparentally inherited. It's inherited through the maternal line, and it is, very importantly, non-recombining. So if you have a bunch of mutations that are happening in the mitochondrial DNA, and then that mitochondrial genome experiences a harmful mutation that kills it, what just happened to the frequency of all the other mutations? It just went down.
You depress them. Yeah.
And the fixation rate for all those other mutations is consequently going to be slower, even though they are not themselves selectable.
Yeah.
And that's background selection. And if you introduce into a non-recombining genome a very strong source of hard selection that only operates 1% of the time, 0.1% of the time, whatever, you're not just affecting that fraction of mutations, you're affecting all of the linked mutations to each of those selectable mutations.
Yeah.
And that reduces the substitution rate.
Yeah. Yeah, and Carter seems to think that the invariance is like the prime evidence for selection, and as he says, like you could have invariance because you could have it because of a low mutation rate, you could have it because of fast genetic drift. There's lots of reasons for invariance. The thing that's a much stronger evidence is the ratios of non-synonymous to synonymous mutations. And so that's to me, that's the lead pipe evidence that the mitochondria is under really strong purifying selection. So for reference, most of the nuclear genome is under fairly weak selection. When you look at the nuclear genome, the DNDS ratios, and that's again the ratio to non-synonymous to synonymous. Non-synonymous mutations means that when you get that mutation, it changes the codon, or it changes what the amino acid that's going to be coded for. When a synonymous mutation happens, you still code for the same amino acid, even though you maybe change the codon. So the idea, right, is that a non-synonymous one is going to change the protein structure because you've changed the amino acid. A synonymous one typically won't change the protein structure because you did not change the amino acid, right? And so you can look at those ratios across the mitochondria, and if there's no selection or very weak selection, then that ratio should be approximately 1. You should see about as many non-synonymous as synonymous mutations if selection is very weak. If selection is very strong, then you should see a lot more synonymous mutations than non-synonymous mutations. And that's exactly what we see. So if you take the average of the DNDS across the mitochondria, it's about 0.14 on a recent paper that I found. That is really strong selection, right? So that's only 14% of the expected non-synonymous mutations are ever seen in the mitochondria. That's direct evidence of very, very strong purifying selection. And this makes perfect sense, right? Like given what the mitochondria does, that it's involved in like aerobic respiration, and all of the proteins for the electron transport chain have to function, not only function in the electron transport chain, but they have to be able to like cooperate with the nuclear genes that also contribute to the electron like they have to crosstalk. It's just very, very strong purifying selection in the mitochondria. And so this idea, and I've never seen a creationist talk about like DNDS ratios, because like to me, there's no other explanation for that pattern outside of purifying selection. I don't know how else you could explain it.
Another way to look at this in the mitochondrial DNA specifically, because mitochondrial DNA is weird in that it is very small, it is very gene dense. So most of the mitochondrial DNA actually is protein coding, unlike the nuclear DNA. And Carter earlier mentioned that control region. the region where we see the elevated mutation rates, that's the region that's non-protein coding. And there have been papers that actually calculated the substitution rate for the different regions of the mitochondrial DNA, right? So for this gene, for that gene, for the control region, for this, whatever, right? Synonymous versus non-synonymous sites. So they also do a DNDS ratio there, same paper.
Yeah.
And the substitution rate in the non-synonymous sites of the coding region is orders of magnitude slower than the control region. Like, we can just directly measure that. And what else is caught? Like, is that region just like magically immune from mutations? Yeah. Like, what is causing that pattern if not the mutations occur equally distributed, but the ones that happen in the genes that change them kill the mitochondria? Like, what's the alternative explanation for that pattern?
Yeah.
I don't know.
Yeah. There really isn't one. And there's like one other kind of funky thing that Carter is doing here is that he's assuming that it always has to be like death or nothing, right? Like that, like either the mitochondrial mutation kills you or it just doesn't affect you at all. He doesn't seem to understand that like if the selection coefficient against it is like 1%, right? Then 1 out of 100 people that have that mutation will suffer from it in one generation. But you give that over several generations and selection will still remove that mutation. Yeah. Right? Because you still have 1 out of 100 people are not reproducing because of that mutation. So it doesn't have to kill all 100 of them. It just has to be detectable enough. It could be 1 out of 1,000. Selection will still eventually remove it. It just takes a little bit longer. And so creationists often get this either all or nothing view of selection, that it's either death or, oh, it's nearly neutral then if it's not immediately killing you, right? And that's just not how selection works.
So he's got this part here that he talks about how, and I don't think we need to play it, it's about a minute and change long. Basically, it's about how in the studies of the mutation rates, he's saying that because you have, as he describes it right, you have these things, again, where hard selection, death, so in the studies, right, of like the pedigree mitochondrial mutation rates, you don't see the effect of the mutations because either those people are rejected from the studies or they're being selected out. The mutations are selected out so fast that they're never.
Yeah.
And like, we don't have to watch him explain this, but like that, there's still selection happening, Rob.
Yeah, it like, goes back to the same thing. Yeah.
The point is that the end goal, right, of a mutation, the thing that's going to happen is either it's going to achieve fixation or it's going to be lost, right? That's what's ultimately going to happen to every mutation that happens in, for us, the mitochondrial genome. If you have some fraction of those mutations that are lost because of strong selection against them, it doesn't matter if you don't see those specific individuals in the pedigree studies, because what we're looking at is the effect of the frequency of mutations at the population level.
Yeah.
And I don't know how to explain that in a different way. It's just, it's not about what mutations an individual has. It's about the frequency in the population. And if you remove individuals, that changes the frequency. And maybe I'm misunderstanding what Rob is saying, but it doesn't seem that complicated as to why this matters to me.
Yeah, no, I think, again, I think he doesn't seem to understand that, like, let's say you observe X number of mutations within a pedigree study and you assume that that's the mutation rate. He doesn't seem to understand why that wouldn't equal the substitution rate, because he doesn't understand that if you could keep tracking that mutation across enough generations in the population, that selection will eventually remove a large proportion of those mutations. They're just not strong enough to get removed in 2 or 3 generations. So again, if the selection coefficient's only 1% against that mutation, it could still persist for upwards of 100 generations before it got removed on average, right? And so this is why you can't think about selection as a fatalistic, it's always going to kill you kind of thing. Selection is a gradual process that removes things over time.
And importantly, as that mutation that has a 1% detrimental effect, right? As that mutation is its frequency is on a trajectory that's different from other mutations in the population, that one mutation in that mitochondria is linked to a bunch of other mutations in the same mitochondrial genome. And now all those other mutations, no matter what's going on with that one bad one, all those other mutations are now going to achieve fixation more slowly than they otherwise would have. And that reduces the substitution rate. It's not about the rare selectable mutations. It's also all of the variation linked to those, whether it's you're dead or you reproduce 1% slower. That's going to reduce the fixation rate of the associated mutations. And
Yeah, and I want to be clear. Like we're not telling stories here. Like
we can talk
the
about
evidence
this.
is literally in the DNDS ratios, right?
Yeah.
So like if we were just telling stories, then you wouldn't be able to look at DNDS ratios and see very obviously that they're not equal to each other. And since they're not equal to each other, the only explanation for that is selection. There's literally no other explanation for that pattern.
Yeah. So I want to play just a short little bit here to wrap up this section on selection.
You're not allowed to participate. So Creationist is completely wrong on that. Plus, mathematically and computationally, you can't reduce that many mutations. You can't remove them without driving your population extinct. So selection doesn't work. All right, the second.
Okay. Anything you want to add to just close the book on selection's not doing the job? Because for my money, with that DNDS ratio and the fact that he said earlier in this video, half of the mitochondrial genome is constant across everybody, I think it's pretty clear that there's very strong selection operating on the mitochondrial genome, and that reduces the substitution rate. But
I think so as well. And I think there's also like other good evidences. If you look at recent papers that have looked at mitochondrial genomes, they constantly find like novel mutations and things like that. And the reason that you can always find singletons is because those are the ones that quickly get removed, and those are the ones that don't stick around. Like we're never finding new common variants or new variants at 20% frequency, right? Like you find these new variants in one family that disappear in a few generations. It's just like that's the way selection works. And for the computational stuff, like again, Carter, like the ball's in your court, bro. Like we've already given you this paper, so either put up or shut up at this point.
Yeah, doing this, like, if you have the selection, then the population's going to die from, like, just, there's the math. You can take, deal with it or ignore it, but it's there. Another one. Thank you. Tad is mad for $2. Crime number 6. Dan is the Pope of genetics. It's me. If you can. I love when creationists do this. I think this is the funniest thing on earth when they're like, this random person on YouTube said this thing that I think is wrong, and here's why they're wrong. Therefore, this entire field is wrong. I think that's hilarious.
You speak for us all, Dan.
Yeah, it's me.
You speak for the whole field.
Yep, specifically me.
Yep.
I do wear hats all the time, but I need to get a funny hat. I need to get a very tall hat if I am to be the Pope of genetics. So Zach, what I have lined up here so that was the first reason of 6, everybody. He has 6 reasons what are going to cause the substitution rate and the mutation rate to diverge from each other. And his point is basically none of those 6 mechanisms work. So we just went through in detail the selection, right? That's the big one. We say selection's happening, that reduces the substitution rate. And he's saying no for all the reasons we just went through and why they're wrong. Now, all the rest are way shorter than that. And Zach, my plan is basically jump to like the 34-minute mark, but just like name check some of the things and why we don't need to talk about them. But if there's anything specific in there you want to talk about, I'm happy to do it. Show the clip and then talk about it if there's anything you highlighted.
I mean, most of the stuff that I have in here is a little nitpicky. So like the drift part that he talks about it. So if you're gonna jump ahead, we'll be jumping past the drift stuff. So
so I just have notes on the drift section, basically saying if you are operating strictly with drift and nothing else, then the mutation rate, the substitution rate are going to be equal, and that's like very basic PopGen math. Nobody thinks that's the case. We don't need to talk about it. Like, just fine, but no one thinks that that's happening.
Yeah, and I'll say that, like, So yeah, I mean, I basically had just like 2 points there. One is
Yeah.
that he says that in large populations, genetic drift doesn't happen, and he says that like very like in this like
Wrong.
very categorically,
Yeah.
and that's just like false.
Yeah.
Drift is always happening unless the population is infinite.
Infinitely large.
That's the only way that drift wouldn't happen. Drift can be very important in in large populations, especially in things like bacteria, which have gigantic populations. However, they all have linked genes, which increases the rates of drift. Selection can increase the rates of drift. So when we talk about background selection and then all those linked things, in effect, what we're doing is we're increasing the rate of drift happening on them. Because selection is not happening directly on them, you're reducing their population size is in effect what you're doing. And so drift and selection are interacting in this way. Anything that's linked to something being under selection is experiencing increased drift because their effective size is getting decreased. So I think he's just totally wrong there. Like genetic drift is important in all population sizes, just in different ways in different population sizes. And then for the mutation rate stuff that you were just mentioning, what's important to note is that the neutral mutation rate equals the substitution rate, and this can be important in parts of the nuclear genome. So there's lots of parts in the nuclear genome that are not functional, that have high mutation rates, and that like virtually match the substitution rates. And we've seen those in lots of studies where those things can match up pretty well.
And importantly, if I just might connect this to the last thing you said, the nuclear genome, except for little bits of the Y and a big chunk of the X, non-recombining.
Yeah.
Or sorry, they're recombining. So you don't have that linkage problem with background selection that you do in the mitochondrial DNA, part of the Y chromosome and part of the X chromosome. So the recombination there eliminates that. So that's why you can have the very minimal effects of drift in the autosomes.
You can have so you can actually see neutral mutations a lot more common in the autosomes because selection is a little bit more focused, because recombination can break up the associations between alleles. We still use those patterns, like patterns of selective sweeps, to help us pick out where selection has been acting in the nuclear genome, which is like a really cool aside. But in general, when we're talking about things like the mitochondria, that, as we said before, is extremely gene dense, there is no neutral mutation rate. Like the mutation rate is always going to be suppressed by background selection. So those are just the things that I wanted to say for that.
All right, then the second thing is he's talking about how you have to remove the males from the studies because mitochondria are inherited through the maternal line, because the sperm don't contribute any mitochondria to the zygote. It's all what's in the egg. So, and he says that's going to have a small effect on the long-term substitution rate versus the mutation rate, but he didn't explicitly say this, but I kind of took away from that part that it would have a smaller effect than he was conceding for selection, basically. It was very minimal effect is what it sounded like he was saying there, which I don't care. I've never like brought that up as a thing. So like the drum that I'm always beating with this is selection, and then to a lesser extent some of the other stuff that he gets to here.
Yeah.
So then number 4 was heteroplasmy, which is where you have mutations in the mitochondria, and then you have variation, right, in the mitochondria within an individual, and you could actually have heteroplasmy inherited if that occurs in the female germline, right?
Yeah.
So because you're not passing one mitochondrial genotype, it would be more than one to the next generation. That could do it. I've literally never brought up heteroplasmy in the context of this debate. Maybe other people have, but like it's just not a thing I focus on in terms of mutation versus substitution rate. So if you have thoughts, I'm happy to hear them.
The only thing that I'll add is like when I've seen heteroplasmy discussed, it's about how selection actually works on the mitochondria. So there's some debate about, since the mitochondria is linked, there's like discussion about things like Mueller's ratchet, like why doesn't the mitochondria undergo Mueller's ratchet? And so some of the theoretical arguments about how it escapes Mueller's ratchet is heteroplasmy, is that you actually just have selection between mitochondria within a cell,
within a single cell,
how you get away from Mueller's ratchet. So I don't know, that's a theoretical argument that I've seen, but that's really the only, from an evolutionary perspective, the only thing I've seen about heteroplasmy.
It's just I have never brought it up in the context of this conversation, so like I don't feel motivated to do any more of it. Then number 5 is one that is important, I think. So number 5, he talks about somatic mutations, mutations in the non-reproductive cell lines in the body. So anything outside of the germline, and he correctly says, you do a blood test, you know, a saliva sample, and you sequence the mitochondria, you're sweeping up mutations that are going to be somatic. They're never going to be passed on through the germline because they are happening in other tissues in the body. And that is true, and that is a problem for creationists trying to calculate this fast mitochondrial mutation rate. And Carter's explanation is, well, you just account for heteroplasmy. If you have a heteroplasmic sample, that's going to be indicative of mutations occurring in other tissues. And if you just reject those samples and only use the homoplasmic samples, that solves the problem. Here's the issue with that. Many of the studies creationists lean on, including the famous Ding 2015 study that Dr. Jeanson bases his entire set of papers on this topic, where he calculates the 6,000-year time to most recent common ancestor. Those studies that creationists lean on explicitly do not account for somatic mutations. And in that particular example with Jeanson, I know this is the case 100% because it was David Neff emailed Ding, the author of that paper, and said, hey, did you filter out somatic mutations from your data sets? And they were like, no, we didn't account for that. Okay, ball game. Like creationists, if you want to find the specific pedigree papers that filter out the somatic mutations, I will stop making that argument. But right now, the ones you lean on don't do it. Like, straight up. So, you gotta fix that.
Yeah, and it's important to note that somatic mitochondrial mutations are much higher, that you see a lot more mutations in somatic cells for the mitochondria than you do in the germline. There was a really recent paper that maybe it's still a preprint, but they were looking at differences in the rates of accumulation in mitochondrial somatic mutations versus germline mutations, and found very strong evidence of purifying selection in the germline mutation rate, not in the somatic line. And so that's a clear indication that and the way that they did that actually is they took a bunch of past studies and they parsed out the somatic from the germline, because they were suspicious that a lot of people had been like keeping them together. Mm-hmm. And when they parsed them out, they were able to say, no, the germline has purifying selection. The somatic cells mostly are neutrally evolving. And so what that means, if you mix them, you're going to get an elevated rate of mutation, right, than what you would expect to see. There's like from a scientist perspective, like not to throw shade on those studies, because it's actually really hard to do, right? Like if you think about sequencing a mitochondria from just a gamete, right? Like that's difficult, because when you take a sample, you're getting a lot of stuff in that sample. As someone that has like done tissue extractions, you've done this sort of stuff before as well. You know, it's very easy to get contaminants from things you don't want, especially when you're sequencing tiny little bitty single celled things, right? So it's hard, and I get it. And the only way to really deal with it is bioinformatically, right? Is to like deal with it statistically.
Right. And I will tell you, talking about small compared to what's in the stuff that I was sampling, or that I was isolating and sequencing in grad school, was bacteriophage genomes. 5.5 kb compared to, and they infect E. coli, which is over 1 million.
Yeah.
So like that ratio is like
it's a needle in a haystack.
You're going to get a lot of contaminated DNA in there, and you have to design your primers really carefully to make sure you're not accidentally amplifying E. coli DNA when you do that. Yeah, it's a challenging thing to do. Yeah. So the last one, which is very minor, and we don't need to spend, I think, any time on it. He spends all of like less than a minute on it. And it's that there are hypervariable sites where you keep hitting the same site over and over again with mutations. So in a short-term study, you're going to see a lot of mutations, but due to saturation at those sites, like if it changes from an A to a T to a G back to an A, then you're going to see fewer mutations over the long term because of that saturation. I've never brought that up as a problem with any of this stuff. I guess other people have, and he's addressing it. But like, sure, I don't care, but that's going to be minor anyway, so like, I don't really care.
Yeah. Yeah, and if you, I mean, and you can account for that statistically, anyways.
Yeah.
If you know the rate of mutation in that region and you know the genetic diversity of that region, then you can estimate whether or not that region is mutationally saturated. It's a pretty straightforward calculation.
So, Zach, that means we get to the fun part now. We're going to jump up to 34 and a half minutes because now he's going to do his concluding bit. And I want to just let this play for about, I apologize, everyone, but it's going to be almost 3 minutes. We're going to let this play, and we're going to let it meander literally and figuratively.
As he meanders.
Right? And he puts a lampshade on it. And so this is the part where we're like, okay, oh, what? So I just want everyone to be ready to appreciate the punchline. Here we go. We're going up to, we're going to go all the way up here to, we're going to do, that was the bit on saturation, I think, was that the figure he showed there. 34. Yeah, this is a good place to start. Okay, here we go, folks.
But one person said it doesn't even matter. And I'm going to say, yeah, it matters a little bit. A little.
All right, that little bit right there is the end of the last thing I just talked about. So,
Little bit. So there's actually 3 things there. Selection removes some of the mutations. Drift does not. The fact that half the mutations occur in males, you have to account for that. That will reduce it a little bit. Heteroplasmy doesn't count. Somatic versus inheritable cell lines don't count because it's more heteroplasmy. But you do get a little bit of mutations at hypervariable sites that will have in some effect. Okay. Yeah, so now I'm at the twisty-turny part of the trail. Perfect timing. Because I'm also getting to a different part in my article and in my presentation to you. You might be thinking that this doesn't go anywhere. Yeah, that's true. You might be thinking that all I'm doing is talking about theory. Yeah, that's true. And assumptions. Yeah, that's true. The conclusions are based on assumptions and theory. I have every reason to question the assumptions. I have every reason to think the assumptions aren't true, and I don't believe the theory. So what am I to do? How do I then explain how long ago Eve lived? Well, I said earlier, the mutation rate is a little too slow for me. But I also said that I don't think that necessarily modern people are a good proxy for all people who have ever lived across the whole world under all environmental or genetic conditions. Okay? And in the Bible, we have something that is profound, something that should have an amazingly huge effect on mutation accumulation rates, and that is lifespans. Old people give more mutations to their children than young people. We can measure that. That has been measured extensively. There is a mutation burden with the age of the parents. We know this. But none of those are done on people who live for biblical lifespans, because no one today is living a biblical lifespan. But something I've talked about, I've done an episode on this show. I have an article on Creation.com, which is in the Journal of Creation, called Patriarchal Drive. That's the idea that the patriarchs after the flood, because they were old when they're having children, they're going to produce children with massive mutation loads. But one patriarch might have a child earlier in life with a low mutation load, but a couple hundred years later, if you have another child, that child is going to have a fantastically huge number of mutations. Therefore, there's no molecular clock. Therefore, we should expect from first principles in the biblical model a period of time where mutation rates were much faster than today. That's what I'm getting at. Patriarchal drive.
That's what I'm getting at.
There you go, folks.
Patriarchal
drive. Now, Zach, I have been a full-time teacher for a decade at this point. You have been an active working scientist much more recently than me. So maybe I'm a little rusty here. Maybe I'm out of practice with this. But this whole video, we've been talking about the mitochondrial time to most recent common ancestor, mitochondrial DNA.
Yeah.
Which is inherited maternally.
Famously, yeah.
And Dr. Rob Carter's explanation for why this mutation rate would be faster in the past is patriarchal drive, as
Because the
in
fathers lived really long. Yeah.
fathers who are hundreds of years old have more mutations in their germline.
Yeah.
I
Maybe he should call it matriarchal drive. I don't know what. Maybe this is a new thing that just dropped. Matriarchal drive.
But the whole point of patriarchal drive is that the male germline replicates throughout the entire life of the male.
Yeah,
the
that's
female germline never replicates.
Yep.
No matter how old you are, those cells are not dividing ever.
Yeah, and like, can you imagine like the rate of aneuploidy at 900 years old?
It
Like,
would
when
be
cells that have
basically
been-
100%.
that are frozen in prophase for 900 years.
Yeah, right. It would be,
That would be wild.
This is, chat, do you see what he just did? He said the explanation for why the mutation rate would be faster in the mitochondria, which is inherited exclusively through mothers, is because the dads were really old in the Bible. Put aside the in the Bible part of that. We'll get to that in a minute. The second thing has nothing to do with the first thing. Mitochondria are not inherited paternally, Rob. What are you doing?
Yeah, I don't know how he came to this, because like so much of this, like you can tell he spent a lot of time on this article, and he spent a lot of time like really thinking through these things. I mean, not all of it was like super logical, but he tried, and he was like really thinking about it. And then it's just here, man, he just threw a Hail Mary. Like he just was like, I honestly, I don't know the answer. But here's the thing that I did some time ago, patriarchal drive. Is that a thing? Is that it? Could that be a thing?
Not in egg cells.
No, he just like didn't carry this thought. Like he didn't talk to anyone before he said this,
It was, in the beginning of that, started off like reasonably okay. Like we have, like he sounded like somebody actually could, like, oh, we have to consider this factor. This factor, not so much. We do have to account for this. That one, that's not actually something we have to worry about because you could just process the data care, right? And that was like, okay. And then there was the part about like, well, I reject the theory. It's like, well, what have we been doing for half an hour, Rob? What's even the point of this exercise if you reject the theory in the first place?
Yep.
And then he goes full, as you said, fan fiction. And it's like, well, this aspect of the Bible affects population genetics, even though they're different. Like it's the males that he's talking about, and it's but what... And now we're just full like we're taking that exit ramp.
It's like science, patriarchal drive.
Yeah, it just I don't know how, because when I watched this video when it came out, like while I was like doing the dishes or whatever, and I'm like, all right, it's a Rob Carter video doing his mitochondrial thing, fine. Yeah. And then today, I did it twice, and the second time I was taking notes, and I'm like, okay, it's wrong, but he's like making like a careful for creationists kind of case. And then I got to this part and I was just like, why did I spend half an hour taking notes on all this? This is ridiculous. This is where it's going to end up, is that guys were 900 years old having kids, and therefore there's more mitochondrial mutations in the past than in the present. Like, what are we doing?
He's ending this book like Stephen King, like he just cannot end a book well, right? It's like such a bad ending to this. Yeah.
I just don't even. So we got Little Pink Bowser here. No, sorry, it skipped. There we go. Little Pink Bowser for $2. Thank you. Go Genetics, patriarchal drive for mutations. What is?
Yeah, and it would also, he never elaborates on what he means by theory. And he also talks about like I don't know if you're gonna play to where he talks about like assumptions. Like he never elaborates what those are and what he means by those things. Because to me, like the theory we've been talking about here is just basic inheritance, and that mutations occur during inheritance, and that's natural selection's a thing that exists that removes mutations, and that we can measure that and we can measure its impact on genomes. Like I'm curious about what part of that he thinks is like theory in the way that I think he's trying to use that word, as in sort of abstract and out there stuff.
Yeah, stuff that we're just like making up.
Right, right. Like I think that's what he means by theory. I think obviously we all know that that's not what theory is in science, but I think that's the way he's trying to use it here.
Yeah.
I don't know what part of this falls under that category, and so like what the quote unquote assumptions are. Again, that like the way inheritance works, the way mutation, like are those assumptions? Those seem to be things that we measure and that we know that these things are operating within biological system. So I don't know what he means here, and the fact that he never defines it to me is a little suspect. And it's a way of him to like kind of weasel out of this. He like got himself into the weeds in this video talking about all these things, and he was like, "Oh crap, I'm in the weeds and I don't have an answer to all this shit." Back up. Let's theory assumptions, and he's just like trying to get out of it. And now he's like throwing patriarchal drive over here. He's just like he's just like flailing, and it's just wild to me.
Yeah, and I want to just pull up two comments here. One, the guys were older, but the women weren't. No, the women were also older, but in the male germline, so you have spermatogonia that are dividing throughout a male's life. And as they're replicating, you're inducing mutations just through replication errors of the DNA. In the female germline, you have a finite source of primary oocytes that arrest in prophase 1 of meiosis before birth. And they only, a very small number of them, will then resume meiosis during the menstrual cycle, after puberty, right? Those cells are not replicating throughout the woman's life, so they're not accumulating mutations the same way you expect to see in the male germline. And he correctly describes this, where you can document that older fathers pass on more mutations to their kids. That is a well-documented thing. The same is not true of older mothers. The thing you have to worry about in older mothers is non-disjunction, because if that tetrad of chromosomes is sitting there in prophase 1 for decades, sometimes they don't separate correctly, and that leads to trisomy or monosomy. And so we would expect a lot of that to happen, but that's not what we're talking about here.
Yeah, and that's also just ignoring like when menopause presumably
Yeah,
starts.
right. You have to
like,
not
like that eventually happens presumably. So you can't reproduce through your entire life in the female line. Anyways.
And then Zach, I appreciated this one. I just want to introduce this into the lexicon. Abraham's ratchet.
That's funny.
I think that's good.
He should own that. Actually, he should write a paper called Abraham's
Ratchet.
I'd
go
read
for
that.
it, Rob. Do it.
Yeah,
do it. We've given you so many good ideas, Rob. I'm still waiting for the Kilimanjaro paper. I'm telling you, have the arc lands on Kilimanjaro. It solves so many problems. So many problems get solved. Seriously. All right. So now we get into the thing that Rob Carter does with like half of his videos, where he gets really philosophical and existential. And that's what we're gonna do here at the end, because this is a Rob Carter thing, and we're gonna watch a little bit of it. So we're gonna jump to here. We're gonna jump up to about there. That'll be fine. And we're just gonna let it go for about a minute and a half to get an idea of where we're coming from here.
Way split. One of my Facebook commenters, I don't remember the exact phrase he used, but he basically said, "Man, the evolution has been treating this like a done deal, and it ain't so." And that's true. It's not so. It's not like we have the answer, therefore the Bible's wrong. Not at all. Take confidence in that. It is not true. The Bible's wrong because they have the answer. The answer depends upon theory. The answer depends upon, therefore, philosophy. The answer depends upon assumptions. And the answer, when you look at it scientifically, all the assumptions are questionable. The philosophy is based on a version of history I disagree with, based on a philosophy I disagree with, naturalism. It's the whole thing is couched in terms of non-finite, non-certain categories with loosey-goosey stuff everywhere. So because of that, I feel perfectly comfortable saying, no, I don't believe that. I think Eve was recent, and here's all the evidence for it. Here's the reasons why I believe that. Here's the things that back it up. And I'm looking at the evolutionary assumptions, and I can question every single one of them. Therefore, Eve could easily have lived 6,000 years ago. There's no scientific proof either way. You got that? You can't use science to know when Eve lived because there are too many variables. You can know that she lived a short time ago because of the mutation rate. You can know that there are a few ways to slow down the mutation rate, but they're not enough to get her to 200,000 years ago. And that's all. That's the answer. Now, this is frustrating.
That's where we can leave it with Rob there.
I agree, Rob. This is frustrating.
This is frustrating to have this conversation year after year. And wind up back at, well, the Bible.
Yeah.
I'm like, okay, I don't accept naturalism. Okay, then what are we doing?
And this is the thing, right? It always comes back to this. Every creationist conversation comes back to this. It's always like, well, wait a second. What do you mean by science? Like we have to take like such a, because you're like, science can't prove this, science can't, and if we look at it scientifically, and then he's like, but I don't agree with naturalism. Well, what do you think science is? Like suddenly it's like we don't even agree on the basic precepts of science anymore, because if you're rejecting naturalism, then that, to me, that means that you accept that there can be a supernatural explanation to something, and you think science investigates that. Like we're in trouble now. Or like now we're in the realm of cuckoo stuff. Like we're not talking about reality anymore.
He's so close. One, he's so close many times in this video to just like the population genetics clicks. But right at this little bit at the end here, he is so close to doing the Todd Wood thing, right? Todd Wood's whole thing. For those of you who don't know Todd Wood, he's young Earth creationist. He's actually like has a degree in evolutionary biology. He very famously a bunch of years ago wrote an article that annoyed every other young Earth creationist. And the article I think was called "Evolution is Not a Theory in Crisis." And the gist of that article and basically his perspective is evolution is robustly supported. It's a robust, well-supported theoretical framework for understanding the world. And I don't accept it because I am a young Earth creationist because of my faith. And the science isn't there yet, but that is the position I hold, and I expect the science to get there eventually, right? And you could disagree with that position, but that is an intellectually consistent position to hold, right? You could say, I accept that the science says this, I believe this for other reasons, I think this is true, and the science will eventually catch up to what I believe to be true. That is almost what Rob did here. But then he went, and you can't actually know anything, and it's all, ugh, and there's like a lot of like, you could see the hand waving, right? Yeah. That's a dishonest position, right? The Todd Wood, the science isn't there yet, but I trust the Bible. That is at least an honest position to take, right? Rob Carter going, we can't know. That is a dishonest position to take. That is, we shouldn't be doing science at all. There's no purpose to any of these endeavors because we can't know.
He also subsequently says we can't know, but we can know it's young. It's like, wait, so can we know or can't we know?
Well, the Bible says it's young, so we know, but then he says because of mutation rates, he says. So like it's
So can we or can't we?
We can know if it's the answer he likes.
It's it's really frustrating because he also just pitches evolution and evolutionary biologists as if we're just theory and assumptions, and like again without ever really fleshing out what he means by that, what these supposed assumptions are, because I guarantee if he were to actually talk through them, we would see that all of those quote unquote supposed assumptions are well established and robust principles of nature, and that we know that that's the way inheritance works. We know this is the way selection works. Like these are all features of biological systems that have been observed. Like these aren't assumptions in the way that and that's the reason he uses the word assumption instead of explaining what he means, right?
Yep. So that was a journey, literally and figuratively, folks. We went on a journey with Dr. Carter in this video, and I hope you enjoyed going on that journey with us.
Yeah, it was a fun ride. It was a fun tromps through the woods.
Yeah, I always appreciate Rob's, where the videos are and why he's doing it the way he's doing it. I cut out the part where he's like, this is a park and they recently built a trail that connects it to this other park and we're going to walk the trail. I cut that part out, but it's nice. It's nice to get that. So that's where we're going to end it tonight, folks. I appreciate all of you being here, watching the two of us nerd out about population genetics and why creationists are bad at it. Zach, I know you're busy in real life, but do you have anything that you're working on in the fake world online that you want to share with anybody?
Yeah. So for those of you that don't know, I recently started a position as a assistant professor, and it's the most work I've ever done in my life. I just have like really been trying to like just stay above water. I get
I have a question. I have a question for you, actually. How
yeah.
soon before class do you finish prepping for class?
Oh, so I think part of my problem is I'm like really, really bad about like I have to be well prepared before I go into a lecture.
Yeah.
So I will just work like 60 hours a day through the weekend to just be prepared for every lecture. That's just like, and maybe I'll get over that like through time, but like the
Yeah.
first semester, it's like I just work
All the
nonstop because
time.
I feel like everything has to be perfect.
But
Um,
but are you doing it like right up until lecture starts? Because all my PowerPoints, and again, I've been doing this for 10 years, all my PowerPoints, if I have class 2 days a week at 3:50 p.m. and all my materials, it's like last modified 3:43 p.m. All of them.
oh yeah. Yeah.
Just
No,
all
so like in general, I try to like have my slides posted ahead of time.
Oh, okay, yeah.
So then I try to not like mess with them too much. I might add like a couple of little things here and there if I find something cool, or if I'm like, oh, I'm going to throw in a couple of like review questions, and sometimes I'll add those in there as well. But yeah, so long story short, been working a ton. However, I do have something that I am working on. I've mentioned this a long time ago, but so next year, which is rapidly approaching, is, for those of you that don't know, the 50th year anniversary of the publication of The Selfish Gene. So
Oh.
it is a pretty big anniversary. Dawkins is also now finished publishing. The Genetic Book of the Dead he said was his last book. And so it seems to me a really good time to make a big video reviewing the legacy of The Selfish Gene, both as a book, as an idea, and then Dawkins as a person, and sort of his impact on both evolution education and maybe just the general skeptic community. It's not like super novel. People have done this before, but I don't know of any evolutionary biologist that has done it. So I want to give an evolutionary biologist
Yeah.
perspective on The Selfish Gene and
Be
what it
that
has
guy.
done for the community kind of thing. So yeah, I feel like it could be fun. Again, given that it's going to be the 50th year anniversary.
Nice.
So
Now I'm looking forward to that. That's going to be great.
yeah.
I've got things coming up. I still have to finish the Genesis Apologetics conference. I've got to finish Winston Ewart. I've got a debate related thing. I'm not doing a debate. It will be a debate intro, response slash commentary slash here's how you should respond to things like this. I think that'll be fun for everybody, but I want to do it in a technical way that is a little bit challenging for my skills. So I haven't quite set it up yet, but I haven't even decided if I'm gonna do it live or record it yet. But it'll be like a time chess match thing, but it'll be like Creationist, me, and you got to keep it time, and I think that'll be fun. I am gonna, Zach, just heads up, I am going to recruit you for the next Galuzza talk at the Genesis Apologetics conference, because he gave 2 talks in that conference. So I'm gonna recruit you, in the usual suspects for that one.
Yeah.
Yeah, I got stuff coming up. I got things. Watch this space. There'll be things. Plus, there's a bunch of like culture war politics stuff that I have thoughts on. Like Discovery Institute just put out a thing. They contributed to, like, somebody put out a thing that Discovery Institute contributed to that has to do with like the criminalization of homelessness, and it's like just really icky and gross, and it's extremely in character for Discovery Institute. So like there's stuff going on.
Yeah, I mean, they've also like completely converted from evolution news to now what, Science and Culture is at the website now.
So it's, what is it? Yeah, I forget what it's called now. I'll pull it up real fast. It is now called Science and Culture Today. Yeah, right.
Yeah.
And
So now they're really leaning into, now it's not just about evolution. It's now we're doing culture stuff, which they've always done, but now they're like kind of owning it.
And that's back to their roots. They were a right-wing think tank before they like grabbed ID as their thing. But for their first few years, their first era was just like fairly generic right-wing theocratic think tank.
Yeah.
So back to their roots, which, it's always good to see people going back to their roots, right? That's nice. Yeah.
Nice to play the classics.
All right, folks. Let's take Rob down here. Thanks so much for hanging out with us tonight. I appreciate all of you. Hope everybody stays healthy. I will see you, if not sooner. I'll have something back here next week. See you next time, everybody. Have a great night.